Good morning and welcome to the first session of the OmicsLogic Data Science program. This program has been designed for the students and faculty at REVA University. In this 1-month program, which is starting today, we will be discussing about OmicsLogic Data Science. Before I start today's session, I want to introduce you to our OmicsLogic mentors. We have Dr. Harpreet Kaur, who is our OmicsLogic trainer and expert bioinformatician, and will guide the hands-on sessions and assist participants with projects and analysis during the program. Her area of specialization is cancer genomics and machine learning. And also we have Dr. Mohit Mazumdar, who is our OmicsLogic project mentor and an expert in big data and computational biology. Before today's session starts, I want to give you an overview of the different resources that you will have access to. So did everyone receive the email that I had sent out yesterday with the access to TBio info and the other details? If yes, then please write the same down in the chat box. And in case you have not received that email, please also let me know. Okay. So this is the first resource that everyone will have access to. This is what we call our organization page. So the organization page is like a classroom where all the members of this group will belong to. And this page serves as that one page where you can go and find the different resources for the program, like courses and projects. And I'll be quickly giving everyone an overview of this. So the first is the forum. So if you see the forum here has different topics that can be created. And the purpose of the forum is to have a discussion beyond these live training sessions, so that when you are doing these courses on your own or running a pipeline and you run into any kind of problem, you can quickly post your problem right here in the forum by creating a topic, which can be done by entering a topic title and entering some details about any of the problem or issues that you're facing and clicking on the submit button here. So here I have added 3 topics. Each day after the session, we post all the session recordings right here under this topic. And here is a guide to getting started with R and Python. So we added you the different tutorials on installation of RStudio and also this blog on Python. You can go through these blogs and these articles here, and also our mentors will guide you throughout this session. Next, you have access to asynchronous courses. So these are all the asynchronous courses that you will have access to. So there are also courses that go over to the next page. These courses are supplementary courses in addition to the live training sessions. So we will guide you on how best to get started with the courses, but you're welcome to try and do as many as you can. And we also have asynchronous projects to help with your learning. And next, what we have is the progress tab. So right now, all members of the group have not done any kind of activity, so everything is 0. The more courses you do, the more pipelines you run, you will see that you get points for each of these activities or tasks. So maybe from next session on, we will be able to see some curves and some different graphs here. For now, it is blank. And each participant will also have their own dedicated profile. So this is just an example of what a profile looks like. You have a profile photo, you have your full name here. So as I was mentioning the asynchronous courses, once you complete these asynchronous courses, you will also receive an automated certificate of completion of this particular course. So that is in addition to this entire program certificate that you would be receiving. This is why you need to make sure that your full name is updated right here. As you see that Kingston has updated his full name. You can also add your social media profiles like LinkedIn, and you will see the different courses you do will keep on adding points to your profile. And we will also be able to see all of your activity right here via your profile. So this will help us to track your progress and help you along the way. And the next resource that you will have access to, which I have also emailed everyone the access, is the server TBio info. And to discuss more about this, our mentors would be guiding you through that. And the other is the Code OmicsLogic platform. And to just quickly add about the code, I'd also sent you a launch code on making sure that you can access all of the courses for free on this platform. And to do that, you just have to go to pricing and click on the starter pack and buy now. And I had sent a code called launch 2020. So it may take some time to go forward with it. And you can definitely do it after today's class. So you just have to type in the code launch 2020 and make sure. And once you apply it, it's going to be 0. You have to enter your card details and then check out. So this is the way where you can actually access the Code OmicsLogic platform. And now to take forward today's session, I would want to invite Dr. Harpreet to take over. Dr. Harpreet, over to you. Thank you, everyone.
Thank you, Deepsa. So, hello, everyone. Let me just share my screen.
Hi everyone and hello Dr. Harpreet. Before you start, I will just have to say a few words. So all of those who have joined, as Deepsha discussed, these are the resources that you will be getting access to. So one of the purpose of today's session is to be able to get all the details of resources that we are providing. So please feel free to ask or ping us in the chat. So you can write down in the chat if you have any issues and if you're facing any difficulties. I got an email that one of the participants and student is not being able to join, and the session is asking for a password and stuff like that. So I have emailed that student. But if you guys have any other issues that you might have faced or facing right now, so please let us know in the chat so that we understand that there is a problem. And for today's session, I mean, the objectives of today's session again will be to first use the resources and get access to the resources and understand the resources. So please check them out, check the links, and then we can proceed to other topics that will cover and will utilize these resources. So over to you, Dr. Harpreet, and thank you.
Thank you, Dr. Mohit. So, hello, everyone. During this 1-month program, we will understand about the various omics data types, data formats, and we will also look into the challenges associated with them. Then we will learn the exploratory data analysis steps required for pre-processing of data preparation and analysis. Next, we will explore the various data analysis tools for large-scale projects, and we will also learn the implementation of various machine learning approaches on biomedical data. And at last, we will look at how we can link these data with the biology to derive biological significance of the data. So in our first session, that is today, we will review various types of data that are used in biomedical research. Some of these will be properties you might be familiar with, like types of the patients, and then disease stages or associated characteristics. But other you might not learn about, maybe the learning first time, like genomics, transcriptomics, and the metagenomics. So all these are collectively called as omics data. But the different with the different file formats, their properties and the biological dynamics they represent significantly. So during this program, we will combine online interactive sessions, self-guided study material, and the practical assignments for an immersive experience that has proven to be effective in our well-known and respected OmicsLogic programs. So let's begin with the history of the genomics data. It's been like 70 years since the discovery of the structure and the function of the DNA. 50 years have been spent since the time sequencing the first human genome, that is around 2003. So in 2015, there are nearly 1,000 genome projects brought a significant number of a whole genome sequences to the research community. By the year 2018, there are nearly 1 lakh whole genomes were sequenced overall. So today there is ever emerging number of the data sets available that help researchers to understand all the biological and cellular processes. So technologies are used to explore the roles and relationship and the actions of various types of molecules that make up the cell of an organism. Omics data can be generated using NGS. So what is we call as high throughput sequencing. NGS omics data can be of various types depending upon the biomolecule it involved. For instance, it can be phenomics that deals with the external phenotype of an organism. Then the genomics that deals with the genetic entities at DNA level or genomic variants. Then third one, that is epigenetics. It deals with the epigenetic modifications like methylation, acetylations of the DNA. Then the transcriptomics that deals with the RNA transcripts or the gene expression of various types of transcripts as well as the isoforms. So overall, these NGS data is widely available for the general public, and this program will allow you to understand what it represents, how to analyze it, and interpret your data analysis results. So I'm pretty sure that every one of you already know about the central dogma of the molecular biology. So just let's look a brief description of it. So it dictates the flow of genetic information from DNA to the protein. So in order to create the proteins from these genes, so first of all, this DNA is transcribed into the RNA via a process that is called transcription. Then this RNA is transformed into the protein by a process that is known as translation. So in other words, protein coding genes, that is RNA, that is it is transcribed from the DNA, that is transcription. So now first our focus will be remain on the transcription. So now let's get an overview, that is splicing. So splicing is a process that removes introns from the sequence. So in a protein coding genes, there are large stretches of non-coding sequences that is called introns. So these introns are spliced out by a process that is known as splicing, and by with the help of an enzymatic complex that is called spliceosome. So finally the parts of the genes that remain within that mature or processed mRNA are called exons. Eventually, these exons are responsible to encode any protein. there are a number of the methods that can quantify and detect mRNA. So can you tell me the method that you know that are used to quantification of the RNA? So you can post your answers in the chat. So name the methods that you know that are used to quantify the mRNA. I hope you must be familiar with the various quantification methods of mRNA. So just post your answers in the chat box. So are you getting my point? Can you name the methods that you know that are used to quantification of mRNA? So I'm not getting any answer. RT-PCR, that's Kavya. That's great. Yeah. Anyone else? Excellent. Kavya? Okay, so if anyone else is answering, so no, let's move it. First is RT-PCR as the Gradhan blotting. So yes, it's the first RT-PCR or reverse transcription. So it amplifies the gene of the interest. Since PCR amplifies only DNA, not RNA. So first of all, in case of the RT-PCR, the RNA is converted back to the DNA prior to the amplification, that is known as cGNA. So this process is called reverse transcription. So in nature, this process is carried out by the retroviruses. As already Kavya told, the next method is Gradhan blotting. So here an RNA sample is first digested into the fragments that are separated via Yell electrophoresis. Then they are transferred to the membrane that can be probed to the test for the presence of hypothesized RNA transcripts. So third one is well known, that is microarray. So it is quite similar to the Gradhan blot, that in terms of it is a hybridization based method. But in contrast to the Gradhan blot, in a microarray, it yields highly detailed information about indivisible genes that have been transcribed. So these signals are then detected, quantified, and used to create digital image of an array. Finally, the digital image is used to identify the transcribed genes. So all these methods that we have discussed so far, that is RT-PCR, Gradhan blot, and microarray, so they can quantify only the predefined transcripts. So what will we do if there is an unknown sequences? So answer to that is NGS RNA-seq. So the NGS transcriptomic sequencing process is a major advance in the study of the gene expression because it allows a snapshot of whole transcriptome rather than just a predefined subset of the genes. Whole transcriptome sequencing provides a comprehensive view of view of a cellular transcription profile at a given biological moment, and it greatly enhances the power of RNA discovery methods. So to study this data, we will learn about processing pipelines that turn a raw reads into a structures data that would be ready for any kind of analysis. Now let's understand about NGS RNA sequencing. So it captures DNA sequences and generate digital data using various steps. For instance, first, the RNA that was extracted from the cell, that is first converted into the cDNA. And then it is sheared and placed into a flow cell. So inside the flow cell, cDNA fragments are amplified using bridge PCR amplification, and then this flow cell is then inserted into a sequencer. So then this sequencer machine uses the image analysis to capture each letter in the flow cell fragment by analyzing visual pattern and converting them into a into the sequence of the 4 letters, that is A, T, C, G. So eventually it will produce something like this, that is called sequence reads. And a sequence reads can be of 30 base pairs to 300 base pairs. So these NGS sequencing data can be stored into various formats, like in the first is FASTQ. So the FASTQ files provide us with the reads from the sequences. It is a text-based file format that stores nucleotide sequence of the reads, and the quality score correspond to each single nucleotide in the FASTQ files. Then the next is FASTA format. So it is also a text-based format that represents DNA or the protein sequence. So typically the FASTA format is actually used for a reference file for a genome. And then third one is GTF file. So it contains genome annotation. For example, this GTF will store the start and end of each exon or intron or any genomic elements. It could be a gene, isoform, etc. Then fourth one is SAM or the BAM files. So SAM indicating sequencing alignment map. That represent the alignment of sequence reads on a reference genome. So it usually starts with a header section, then followed by a alignment information as tab separated line for each of the read. So BAM files almost store the same information that of SAM files. The only in case of the BAM file, the format is different. That is a binary format. So these are different file formats where NGS data is stored. So then to understand why other steps are needed in the RNA-seq analysis pipeline, now let's look at the expression of the genes, how data can be used to study this. So this is like a reference genome where you have gene A, gene B, gene C. So not everything is going to be transcribed. For example, here we can see that only gene A and gene C are getting transcribed. So the product of gene A and gene C is become a RNA that will be shattered into the pieces. And then to these pieces, the adapters are aligned at the beginning and at the end of each fragment. So then this nucleotide information is amplified using PCR amplification. And then finally we got the reads that will be read by the sequencers. So now just let's look how we can perform RNA-seq analysis. So for any RNA-seq analysis, there are 3 major steps, that is pre-processing, mapping, and then quantification of the gene expression label. So at T-Bioinfo platform server, you will have, every one of you must have the access of T-Bioinfo server. So on T-Bioinfo server, each step is actually indicated by different color codes. So for instance, pre-processing steps are actually encoded by the blue color. Then the mapping steps are actually coded by this red color. And then the quantification of the gene expression will be coded by this green color. And at last, the any kind of analysis like differential gene expression analysis will be encoded by orange color. So now let's look at how Ramesh, I have a question, that is RNA-seq having T. What do you mean by T?
Madam, madam, actually, I am Dr. Ramesh here. So in RNA sequencing, it is saying T, means that is not RNA sequence, that is you can say that is complementary of DNA.
Yeah, that's a complementary DNA, that is cDNA.
Okay, so.
Because in case of the RNA-seq, the RNA is first actually is converted into the cDNA. because you
So
cannot
whatever sequence, madam, actually, whatever RNA sequencing it is taking, we are going to make a suppose complementary DNA, and this data means whatever sequencing data it is coming, that will having a, you can say it's a cDNA, not a RNA.
but it is derived from that RNA, right?
Yeah.
Yeah, it is derived from that RNA actually.
So we will
That's why
say
we call
RNA
it.
sequence.
Yeah, exactly, because it's the source of the source of whole this overall this process is actually the RNA, because you cannot amplify RNA as it is. So to amplify that RNA, we convert it into the cDNA.
Okay. Okay, madam.
Okay. So now let's understand these steps one by one. So in the pre-processing, we usually do two steps. That is, first we remove the adapters that we have added at the end of the sequencing reads. And then we also remove the any PCR duplicates. So this is important because PCR amplification is not uniform across all the reads. So then we take these cleaned up reads and then we map them onto a reference genome. So on a here we use the FASTA files for the sequencing of the genome, and a GTF file is used for the annotation. And based on the quality of the annotation, we can use various strategies for the amplification. So, and we can also use various different algorithms for the mapping that I will let you know when we will perform this demo on how we can perform RNA-seq analysis. So once this mapping is completed, then we have to quantify the expression gene expression label that actually represent in particular in a number or a value. So now let's understand RNA-seq analysis using a demo pipeline that is available at TBioInfo platform. So before I will go details into for the demo, I just let you know the different steps. So here, like the first section, as I have already told you, on TBioInfo server, you will find color-coded sections. So under this pre-processing step is coded by the blue color. So even at the start of every single step, it is there are different methods or different steps or different algorithms available for every single step. So pre-processing is indicated by blue color. Then the mapping is indicated by red color. And then this to quantify the gene expression, it indicated by the blue color. And if you want to perform any kind of analysis like differential gene expression analysis, it will be indicated by orange color. Finally, you can end off this pipeline. So now let me take you to the TBioInfo platform server. So if every one of you have account, you can just perform this demonstration with me. So just log into your this TBioInfo server. So just let me know if you able to log into your TBioInfo server. Just write your yes or there in the chat box. So Vipsa, beside you also post the this URL for this server so that they can easily log into this server.
Sure.
So just write write yes or no or there in the this chat box so that I will understand that you are with me.
Dr. Arpit, can you show them how to log in? using like
Okay,
start
okay.
from the beginning.
Okay, okay. Let me just log out. Okay, so once you are on this server, then you will find here login.
And just to add to it, the email subject line is as I have posted it in the chat.
Just I have to, I have to find the login details. Just give me a second. So I'm just stopping my screen sharing. Just give me a second.
So if
Yeah, and as Deepsha was mentioning, go ahead, Deepsha.
Yes, so I had sent this to everyone yesterday. So please let us know in the chat box. So if you do not let us know that you're following through the sessions, we will not understand whether you can follow this class, whether you have the access. So please let us know right now in this session, as all of us are here and we are talking to each other. Put down in yes or no whether you have this access or not. Because if you do not have the access, then as we progress through the sessions, it would become difficult to follow up, right? So if we can make sure that everybody has the access today, so the next classes would be much more smoother. So how many of you have the access to TBioInfo or can find that email which I have sent to you yesterday evening? Can you please put down in yes in the chat box?
And Dr. Ramesh, if you can also announce this, if you're there, and if you can see your...
Hello sir, actually. First of all, I'm trying actually, but I did not get, so I don't know. Some students, they're saying yes.
Okay.
No, somebody having problem, okay. So I'm also trying simultaneously actually, but even in my case also it is not getting login.
So let's take this time. I mean, search for this email. You can use this subject line, OmicsLogic Data Science, and search in your inbox, and you will find the email. And
Yes.
in that email, you will find the details.
So for everyone, I had sent it yesterday. So please search with that email. Okay, this is again the subject line. Please search with the subject line. You should receive an email from marketing@pine.bio. You can also search.
Yeah, that one. Excuse me. That one I deleted by mistake. I don't know.
Okay, Dr. Ramesh, I can send it to you right again.
Yeah, correct. Actually, yesterday I have seen night, and I told to everyone everyone I think so you received mail, and but I got this link from some. Yeah, okay, fine, thank you.
Okay, so just here, I just let you know how you can, if you have your details, so how you can log into the system, just enter your email ID.
And whosoever has been able to get login and been able to log in, please tell us that you have logged in. So if you have not, and then also you have to let us know what problem that you are facing, because unless you know, it will be difficult for you to follow the classes, because it's important to have access to these online platforms that we will be using very frequently. So please try this out right now and figure it out. If you're having problems, let us know.
Yeah, hello, a student, we have to give response, okay? So this session should be complete fruitful. Okay.
Yeah, it is an interactive session and we are asking again and again, and that is the reason, because we want you to at least write in the chat.
Yeah, I think so. Most of somebody is saying no. I don't know.
Yeah, so I think
Preeti.
Yeah, let me just clear this up. So if you do not see the email, it is because I will forward you the email again. For everybody who has said no, I will forward you the email again so that you receive it. Okay.
Yeah, okay, madam. Okay, madam.
Great.
Yeah, just again.
Yes,
Even,
yes.
even I'm not able to know.
Yes, Dr. Ramesh, I'll also send it to you again. I know this is just the first session, so it's okay. Let us take it slow. Everybody will have access. So right now, Boishaki, you should be receiving the access again. I'm just emailing you again, the access, and I'll follow it up with everybody else who has said no.
I think Meghna, Mahati.
Yes.
Samyukta. Lot of people, I
Has
think.
access actually, so I think they probably missed out on the email. So I'm just sending it to everyone again.
And the email address that is that in which you
Marketing
have
and find
received
out bio. Yeah.
no, the email address that they have received is their academic email address.
reva.edu.
Yeah. So that is if
reva.edu.in.
yes,
Yes.
if you're looking in your personal inbox, you will not get it. So you have to look into your REVA inbox, and then you will be able to get it.
So if any one of you able to log in, just let me know that as well.
So there are a lot of people who were not able to log in. So
I think
many
a lot
have
of
received.
people. Yeah, we can wait for that.
Yeah,
At
we
least
can
it's
wait.
a session.
Yeah. Okay. So everybody please check your REVA email ID, and I'll be forwarding everyone the email again, like I forwarded it to Boishaki right now. So, okay.
And Dr. Ramesh, I have shared your username and password privately to you. So please try to log in if you can log in or not.
No,
Let me
also
know.
for myself also, but it's not login actually. I
Yeah,
was
I
trying
have
to.
used the username and password I have given, I've pasted in the chat to you.
Okay, yeah,
And
I got it. Okay, I'll
so
put this one.
please, yeah, please make sure there is no space when you put the password. No, usually there is a space
You
that
just
is.
copy and the paste. I guess that's the
Yeah,
best
okay.
way. Yeah.
Okay.
So like here,
Next up is
just
Shreyoshi Banerjee. So Shreyoshi, just check your email right now, your reva.edu.in, and you should have the email sent to you.
Yeah, now it is open now.
It's
I
okay.
got
that's
access.
great.
Great.
That's great.
Okay. So Shreyoshi, you should receive it. Just give it a minute. I just sent it to you. And let me know once you do. Okay, so Amagha already has it. So Kavya is next up. Okay, Kavya, I'm sending it to you again.
So meanwhile, I just let you know what we are going to do. It's like we are starting from those raw reads that is in FASTQ files. So from those raw reads, we finally how we can actually generate a gene expression table. So like on a sequencing raw reads, can we perform any kind of analysis? I think we cannot derive any biological interpretation from raw reads. So here what we are going to do, that we are going to generate a gene expression table that would be a very structured data from those raw reads. So there will it will involve various steps. I think some of are not received any.
So, Dr. Harpreet, will look into the chat, please.
It is
We will.
just looking. Yeah, I'm just sending
Yeah,
it to
you please
everyone
carry
again.
on with the
Yeah.
session. We will look into the chat.
Okay.
So Deepsha will be sending everyone again, and we are also sharing. But the mail has gone to everyone. So please, for now on, add marketing@find.bio. Just I'm writing it down to your address book or something, so that you get mails and in your inbox. So we have this confirmation that the mail has been received by all of you in your inbox. So please have a look at your emails, because we are going to email most of the things to you that you should be receiving.
So here your username most probably your mail ID. So should I go ahead with the session if some of you have already able to log in? So just let me know how many of you are able to log in.
Yes, Dr. Harpreet, please go ahead. I mean, this session is
Okay,
also recorded, so you guys can
okay,
always look back and see what had happened and repeat it.
okay. So once you log into this system, you will find here different sections like NGS data, mass spec, or structural biology data integration. So under the NGS data, here will be a subsection that is transcriptomics. So in the transcriptomics, there is the first option that you are getting, that is RNA-seq or /chip parallel analysis of NGS and microarray data. So in the site, you will find a drop down. So if you click on a drop down, you will find here 3 different demo pipelines. That is demo RNA-seq cell line project, then demo RNA-seq Angelman project, and then demo RNA-seq PDX project. So now let's begin how we can perform any demo. So let's start with the first, that is demo RNA-seq cell line project. So once you click on that, then you will find the this complete pipeline workflow here. So as this is a demo pipeline, so here we are not on the start, we have already uploading the files. So as this is a demo pipeline, we are not providing any, we need not to provide any input file from outside. So as we have already provided the required input files. So in samples.svl, it contains the FASTQ file, URL of the FASTQ files. Then we are also providing the reference genome. So here we are just providing our FASTQ file that contain raw reads and the reference genome file. So once you click on the start, then you are actually allowing that you are uploading your files. So once you upload your files, then under the pre-processing step, first option that is getting highlighted, you will find here Streamomatic. So what is Streamomatic is doing? As I have already told you, during the pre-processing, we are removing the adapters. So Streamomatic is actually remove the adapters or any technical sequences. For instance, pink color indicated any technical sequences, and adapter it indicating it is indicated by the blue color sequences. So eventually the Streamomatic removes any technical sequence as well as it removing the adapters. Once you remove the adapters, then the next options that is getting highlighted, that is PCR clean. So it will remove any PCR duplicates from the reads. So every one of you who are with me, so are you following these steps? So once we have performed pre-processing of our data, next thing that we are going to do, that we are, after removing these duplicates as well as adapters, finally we are going to perform mapping. That is, we are aligning the raw reads on a reference genome. So there are different number of the algorithm that can be explored. So you can use different algorithm depending upon your study design. So here we are using HiSet. So what it doing, that it aligning your reads on a reference genome. So then the next step, that is cufflinks. It will, as it indicated by this purple color, what it going to do, that it going to generate isoforms. So on clicking on OK, we are performing the steps, that is isoform construction. That the next option that is getting highlighted, that is cuff merge. So cuff merge is under the GTF processing. So it will annotate your transcripts and it will merge the different exons together. And then the next step, that is after GTF processing, the Butai2T, it will map your reads on a transcripts. So with this, you will get the transcripts. Finally, under the expression label, or that it will quantify once your reads get aligned on a reference genome. So finally, you are going to quantify those reads that are correspond to a specific gene or a specific gene expression transcript. So this RSCM, with the RSCM X table, you are going to generate your gene expression table. So it will accurately quantify your transcripts from the RNA-seq data without reference genome. So here you will you can perform various kind of you can quantify your data into different formats. You can quantify data in form of the counts. Then there are the normalized values like FPKM and RPKM. So I will let you know what is FPKM. So here you are actually quantifying your data in form of a table. The next step that is going to highlight here, that is DSEC2 under the differential gene expression analysis. As here our aim is to only to produce a gene expression table, so I am going to skip this DSEC step. So then I am getting the next option, that is end. So with the end, I will with this end I am completing my pipeline. So if any one of you able to perform this demo with me, so just let me know. Just write yes or no in the chat box so that I will understand if any one of you able to perform this to run this pipeline to generate a gene expression table from a raw reads. So even if some of you not able to do this, you will find the video of this session again, and you can perform and you can run your pipeline yourself when you will get time, once you're able to log into this. So any one of you able to perform run this pipeline, just write yes. Otherwise I'm repeating these steps. First of all, with the start we are uploading a file. Then under the pre-processing steps, then is Trimomatic, that will remove the adapters from the raw reads. Then PCR clean, it will remove any PCR duplicates. Then with the HiSet, we are performing aligning of the reads on a reference genome. Then the cufflinks, the next step is cufflink. It will generate the isoform, that is isoform construction. Then cuffmers, it will perform the GTF annotation, that is for the GTF merging. It will merge your exons. Then the Butai2T, it will map your reads on a transcripts. And with RSCM expression table, you will able to quantify the gene expression table, and you quantify your RNA expression. So with end, you are completing this pipeline. Once you're done with this, then you will find here various files under the output files. So in the download option, once you click on this, you will find different files, like here the expression gene FPKM not filtered. So this file contain expression of all the genes in terms of FPKM, and expression genes FPKM, it will contain all the genes, but it will remove the genes whose when there is no much variance in the expression of that particular gene among different sample. Then you will find the expression of your isoforms in these two files, that is isoform FPKM and isoform FPKM not filtered. Then this is how you will contain various kinds of output, and this cell line clinical type, it will contain the clinical information of those particular samples. And this mapping stat tables will actually indicate the quality of your mapping. So if we just go to this, I'm going to this particular table, that is expression gene FPKM. So any one of you able to run this pipeline, just let me know. So Dr. Ramesh, if you able to run this pipeline.
Hello, ma'am. It's not coming for me. Actually, I was looking your presentation, so at that time again I went say yes.
So
Okay.
let's just repeat it and please see it
Okay.
and then try
Okay, I
now.
can
I
repeat
mean,
it. Okay, just just.
No, they already is recorded now, so I will see actually because I was looking here for your presentation. That I when I again went actually I missed.
so it will
Okay, okay.
not
So
take time, Dr. Ramesh.
much
I
time.
think just
It will take
steps.
a just
Yeah,
a
just
few minutes, just
Oh,
for
okay,
one or
okay,
two.
okay,
let's.
sir.
So if you are on this page, so let me
Yeah.
start with the, okay. So let
So
me just
first,
start with this area of analysis. So under the area of analysis, under the NGS data, this is a transcription option. So under the transcriptomics section, first pipeline. So here you will find a drop down. So under the drop down, let's go with the first demo, that is RNA-seq cell line. So you can perform any kind of, I mean, for other demo pipeline. So I am just going with the first, that is RNA-seq cell line project. So here we are using cell line theta. So if you click on this demo with the RNA-seq cell line, so then you will find this option. Are you able to find this?
yeah, correct.
Okay, okay. So with the start, we are actually uploading our raw read data that is in the FASTQ file. So
Yeah,
usually we provide in the form of samples.svl. So it's like, it's very difficult to upload your files as such. So we will provide always the URL. So you need to provide only the URL of the FASTQ file.
okay.
So that is in SVL table. So once you've done this, okay, so you are actually uploading the data, as this is all demo pipeline, so we have already uploaded these files. So here you need not to upload any file. With the start, we are uploading this file. Then the trigonometric step, it will remove the adapters from the raw reads and any technical sequences that is associated with the read. So we are removing all that all those things with this step, trigonometric. So once we able to perform trigonometric, then we are going with the PCR clean. It will remove any PCR duplicates. So you just click on PCR duplicates, then with the OK, you are performing this step. Once you able to do this, then the this is under the mapping. The next step is mapping. That is, you are going to align your reads on a reference genome. So the next option that is automatically getting highlighted is high set. So you just click on high set.
Yeah, correct.
So it will align your reads on a reference genome. The next option that is getting highlighted,
Reference is what we are taking here. How will come to know which one reference?
that is reference genome to that particular cell line. Suppose we are using any human sample. So in that case, we using Homo sapien. I mean, yeah,
Normal.
that is.
One is normal, another one is cancerous cell.
So no.
Like,
Reference genome is always like if to a particular species. Like for the human, so there are
So
the different reference genomes available for like even latest one is HD38, right? So if you have bit idea, so it's like a the reference genome to a species, the normal, I
so
mean the normal.
generally we'll say the normal cell line means normal genome, and we can say suppose cancerous cell because of difference in genome sequence, somewhere mutation something is there, no? So we can say.
Exactly.
So, Dr. Harpreet, if I may interrupt. So, Dr. Ramesh,
Okay.
Yeah.
what we are trying to do here is first we are generating reads from patients, like you said, a normal patient and a healthy patient and a
Correct.
sick patient, right? A person that has this cancer and person who does not have this cancer. So, we generated, we collected samples from their tissue, and after collecting the samples, we sequenced them, right? Now, normal and healthy, and the one that has
Cancerous
cancer
person.
or cancerous, yeah. So what we need to do is now to know that where these reads are, aligning to the genome in general. Like you don't know, right? When you get reads, it's small reads, and now you have to make it into a genome, right, or a transcriptome. So for that there is reference. So there is a when you're doing an alignment, so there has to be an alignment with another sequence, right?
Correct.
So we have a full sequence of the genome, which is what Dr. Harpreet was saying, that GR37 and GR38 are the two reference genomes. So right now it is the GR38. So that reference genome has all the information about human proteins, genes, all that information. So that when you align your transcript, you know which is matching to which.
Correct.
Yeah, that's the reference genome. And in this pipeline, the reference genome is already selected because we are working on human genome. So the reference genome is again human. So if you are doing any plant experiment, if you're working on Arabidopsis, you have to align it with Arabidopsis genome. Likewise, if you're doing mouse, you have to align with mouse genome. So it's just like that. So it depends on the experiment that we are doing.
So here we are doing alignment, means one is reference, another one is cancerous.
No, cancerous normal, like person. If you have sequenced your genome, for example, your genome is sequenced. So now we have to know which are the genes, which are the proteins, where your reads are aligning. So any genome that you sequence, you have to align it with some reference genome, right? To know where
so I'm saying actually here mine sequence, we are going to align with a reference sequence, whatever already it is available.
Which is the annotated one, yes.
Correct.
Yeah. So that is the reference genome, not like normal or cancerous. Whatever normal cancerous, you have to align it with reference genome. It does not matter.
Yeah, a standard one. That is a standard. Already they have annotated complete annotation. Already they have done.
Yeah, I mean, whatever level of annotation they have been able to do, that is there. I mean, so model, like you can say model, that is a model genome.
Model. Okay, sorry.
Yeah. So we compare it from there. Is it clear
now? Okay, now it's clear.
Okay, great.
Thank you, Dr. Mohit, for clarifications. And then the next step, after you align your reads on a reference genome, then next is cufflinks. It is used to the isoform constructions. So then if you click on the cufflinks, so then on OK, we are ultimately completing the isoform constructions. Then the next option that is getting highlighted, that is under GTF processing. So as already we have told you that GTF is actually used for the annotation. So cuff merge, what it will do, it will merge the different exons. Once it got the different exons, so it will merge them together. So once you able to done this cuff merge, then the next option that is getting highlighted, that is Butai 2T. So here, as we are generating the finally the transcripts, so this Butai 2T, it will align the finally these genomes on a transcriptome. So once you able to align your reads and sequences on a transcriptome, finally the next option that is getting highlighted, that is RSCM expression table. With this, you able to produce a gene expression table. So you're able to quantify all those reads actually. So here you can, you will get the values in form of the cons. It could be a cons. It could be some more normalized like FPKM, RPKM. So I will explain you in the just the next slide what is the FPKM, RPKM. These like they are quantified values in the normalized form. So then the option is getting highlighted, that is D step. As here we don't want to perform differential gene expression analysis, so we can skip this step at least in this pipeline. So we are going to end with this end. We are actually completing our pipeline. So eventually the final step that we have performed was RSM expression table. So we are going to generate a gene expression table.
So DCQ to why not we did not, this one also is highlighted na? after
Yeah. Here our aim is to we can do that, but here our aim is to only to generate a gene expression table only. So we
okay.
can perform any kind of analysis depending on our desired analysis, right? It doesn't need only the differential gene expression analysis. So that why we need to what kind of analysis we need to perform. So that's the other thing. So here we want to produce a gene expression table so that we can perform various kind of analysis. So you cannot perform any kind of analysis on raw reads, right? So finally we able to generate the, so under the output files, you will find various kind of outputs. So like here, different expression for the genes, expression for the isoforms. So you will also get the clinical data for this particular file. Apart from mapping, static will actually indicate the quality of your sequence alignment and mapping. So I am here getting the table that I'm going to show you, that is this expression gene FPKM values. A second. So I am going to paste this in a Excel so you can.
So, Dr. Ramesh, did you get the output files?
Yeah,
As
this
well?
sir, I downloaded that one. It came in that a notepad form.
Okay,
Yes,
so yeah.
and you can open
So
it in Excel.
yeah. So this is like, finally, I have just pasted in the Excel so that we can easily understand. So this is like the gene expression table we got from those raw reads. So here is very well structured data finally. Here, especially the columns are representing the samples, and the rows are actually representing the genes or the transcripts. So this is of the table that you will get end of while running that pipeline. So we are eventually be able to produce a very well structured gene expression table from unstructured read set files. So now we can perform any kind of analysis like here. So if you look at here, so in the first column, or you will get like different ensemble IDs for particular genes, like how you will find the whether it's a transcript or a gene. So like this ID, I'm just highlighting it. Let me just, so here like some IDs like ENSG, then particular some number. So ENS, what it indicating? It indicating the ensemble IDs. So then the G. So what the G is indicating that this particular ensemble IDs belong to a gene. So if instead of G, it would be like, it could be T. So T is for transcript. So this is like the different ensemble IDs for each of these genes. And these are, so finally we have a gene expression data table where we have the samples in the row and genes in the, samples in the columns and genes in the row. So now you can even look into that. Suppose I am just collecting this one gene and I want to look into what kind of the gene I'm getting. So we can generate. So like here we can produce any war plot for this particular gene in different samples. So this war, I'm just generating a single gene expression. I'm taking only a single gene. So I can look into them that how it looks finally. I can perform any kind of analysis with this data. So like here with this gene, we can look into how it is getting expressed in different kind of the samples. So with this, finally the table that we have obtained, we can perform any kind of analysis. So this is how we are able to produce a simple gene expression table from unstructured raw reads.
So Dr.
I
Ramesh,
hope.
is this clear?
Yeah.
Okay. So now let me go back to the presentation.
So usually this is a really intensive process and you need servers and high computational power to be able to do that. But you saw how easily you can do it on the Bionfo platform because you have it everything on the cloud. So running from millions of reads, so this is in megabytes and gigabytes of data, and then you getting to a table of expression is one of the important things, because once you see the table of expression, then you learn about the protein expression, right? And then you can understand whether in this sample this has less expression and this has more expression, means that there must be something, you know, different, and something different is happening in cancer versus normal. So there are different samples, there are different gene expressions. So it's a
What is
simple
that
table.
gene, sir? What is that gene actually? They have only ID number, no? This is
Yeah, so
simple
you
ID.
have to now annotate it. So you can search with this ID and you will know which gene it is. So Harpreet,
Okay.
can you
I
just
think,
search
yeah,
once
yeah.
and
Okay. So
yeah, and
suppose I'm going with this particular. So this is like I have already told you. This is these are ensemble ID. So otherwise there are different tools with which you can annotate which particular gene. So I'm just like here I'm taking a example of this one. So I will just search I am just pasting. So here I will get the information. So what it indicating, this is ensemble is an actually an very intensive software that that is like it is a database that contain annotation annotation of all those genes transcript. So finally when I have just click on this, so I have find this gene summary. This is trans and 6 gene. So you can get the information like this ensemble ID belongs to which particular gene. So like in the, this is an ensemble. So here we will find the information like this is uh, uh, this particular gene is actually TSPAN6, which is tetraspanin 6. Like so similarly we will get the we can get the annotation for all those gene id ensemble gene id. So this is just I have given you just one example. So this is like finally you have the genes here just represented in terms of the ensemble id, as ensemble ids are usually uniform, I mean. So that's why sometimes why exactly these algorithms exactly producing the ensemble id instead of the gene names, with time actually sometimes gene names are getting modified. So that's why just to make something uniform so that it ensemble id is always a unique ids, ensemble as well as gene ids. So those are the unique. So names some can be modified. That's why here we are just our this particular resource is actually producing the gene expression table that represents those unique ids rather to the something that is actually getting. So you can annotate your ensemble ids eventually. So here like if we have like these 3 samples belong to the cancer and these could be the normal, so we can understand, we can perform any kind of analysis on this data. I hope Dr. Ramesh it's clear to you.
Yeah, it's okay. I got it. I search in Google also, madam.
Okay. So
and
these are
also for the
ensemble
students,
IDs.
Yeah.
let's ask the students if they are
Yeah,
having.
from
So
a
it is
student
clear.
actually.
Okay.
Dr.
Yeah.
I'm learning just for my enthusiast nature. Otherwise, no need
Yes,
actually.
yes, absolutely. Everyone should learn, and I think the students have to learn as well. And Dr. Ramesh will be helping, I guess, the students
No, I
if
will
in case
help.
they
If
need to.
I definitely, if I'm understanding, I will help it out the way actually. Suppose after 1 month, suppose they are going to ask again, I will keep always
Yeah,
definitely I will support the.
sure.
Okay.
If I may
So a
just
student,
add,
everyone kill you.
Dr. Mohit, if I may
Hello.
add, yeah. Dr. Ramesh, can you hear me?
Yes, madam.
Yes. So some of the students just confirmed that probably the email was put under quarantine for everyone. So that is why most of the people did not receive the email. So I had emailed everyone around 10 p.m. last night. So
Okay.
yeah, so that is why. So I will be forwarding it to anybody else who has not received it. So just let me know, because the emails are already made, and at the end of the session, I will just explain what are the different things again, which I sent in the email, so that everybody knows the different accesses that are there. Okay, and
Okay.
over to you, Paul. Thank you, Dr. Harpreet.
Oh, thank you, Lisa. So as I have already told you the few terms I have used, that is FPKM value, RPKM or the TPM. So let's understand exactly what it indicated. So FPKM is fragments per kilobase per million of the mapped reads. So it's like your number of the reads that mapped on a gene, then divided by the total number of the mapped reads in the millions, then also divided with the gene length in the kilo. So this is how you can compute your number of the reads. So apart from the FPKM, there is RPKM. So the difference between FPK and RPKM is like FPKM is for the paired end reads. I think everybody maybe you know that there is pair end sequencing and it's single end sequencing. So in case of the single end sequencing, the same thing is computed as RPKM. So and FPKM is the for the parent. As in the parent, instead of the reads, we are using a fragment that actually contain the reads from both the ends, that fragments. So here we are actually computing the fragments, and in case of the RPKM, we are actually dealing with the reads. So it's like number of the reads mapped on a reference genome, then divided by the total number of the mapped reads in millions, and divided by gene length in the kilobase. Apart from that, there is another term, that is TPM. It will indicate the transcripts per million reads. So these are just the otherwise, as you get the data in the ponds, so this is like a more normalized quantification of those reads. So I hope everyone able to get this point. So this is just the gene expression table that we got, and here we are getting the gene expression form in terms of FPKM, the TPM. So every single table, as I have already told you, it represents the samples. And
Madam, approx we got approx 26,000.
Yeah, exactly. So finally, to to filter out this data, and finally how we can process these data, that we will discuss in our upcoming sessions. So their normalization, their quantile normalization, so those kind of things we will discuss in our upcoming session. So now as we got this structures data, which is very easy to analyze in comparison to the raw read, you cannot perform any kind of analysis on that, right? So finally, we able to quantify all the gene expression table in a structures form. So this is how you actually can understand then what are the outliers. So we will discuss all these things in detail in the upcoming session. I'm just here just letting you know. So once we have this table, we can like I have plot a war plot for a single gene. So like similarly, you can perform various kinds of box plots for all those genes and can analyze what are the outliers, right? So here, like we all we know that what this box plot represent. So it is like it indicate the median value, and it indicating the upper quartile, and the lower part of the box always indicate the lower quartile. So apart from that, then this is a minimum and maximum values. So if any value or any gene is any sample that is beyond this minimum and the max value, we consider them as outliers, like here. So it's like it's minimum or this is beyond its minimum and the maximum value. So this is how you can identify which samples are actually representing the outlier. So to further identify a gene as a significant marker, so you have to delete all those outliers. So these kind of analysis you can perform on this particular, once you got this particular gene expression table. So then you can understand which gene is getting upregulated or downregulated in different samples. So then before, as if you look at that table, your value is varying from very, like it could be varying from minimum to the maximum, very large extent. There must be a very large variation. So to handle that variation, one of the method is like log scale transformation. So it will, like, you always transform your first, once you get your FPKM value, you usually transform those values, so that it's not like one value is like in samples it's varying to 5 and other is like 1000 or 10,000. So it's like it makes a value with in a minimum range, like it could be 1 or 20, right? So that's why we perform log scale transformation. That is that will help ultimately. Those are the pre-processing steps, and we will discuss all these steps in our upcoming session. So all the things that I have discussed so far, the topics that I have discussed, and even the detail, you can, all those details you can get we have this one program course at our educational platform, that is Transcriptomics 1. You will find all the details regarding all these pre-processing and RNA-seq analysis in much detail at Transcriptomics courses. So I would request every one of you, if you just go through these courses to understand everything in a more detail. So beside, if you want to understand transcriptomics, we have 3 more courses like Transcriptomics 2, Transcriptomics 3, and Transcriptomics 4. In Transcriptomics 2, you will get the information regarding how you can perform various kind of pre-processing and post-processing steps. The Transcriptomics 3s belong to the where you can how you can apply different machine learning approaches like supervised or unsupervised. Transcriptomics 4 deals with the single cell genomics. So let me pause for a while. So if you have any questions till now, before heading to the next part.
Yeah, student, you can ask question. Otherwise,
you post your yeah, you can post your questions in the chat box.
Akshita, Anaga, Anirudh, everything okay?
And Dr. Ramesh, there's an opportunity to post in the forum also. So let them learn this, and they can post anytime.
So if you face any problem during this course, I mean during this course, and then when you are completing your course, so you can post your questions in the forum as well. So now let's move to the next section, that is genomics. So whenever you listen a term, we listen this term genomics, what comes to our mind? So to me, like it's genes, it's DNA or ATGC codes. So these genes pass from cell to cell. So there are nearly 3 billion letters in a total. So that encodes nearly 22,000 proteins. Genes are made up of a biochemical entity that is called a DNA. In short, we also call this deoxyribonucleic acid. So it is a double-stranded DNA helix that is too long and thin strand twisted around each other like a spiral staircase. So in the DNA, backbone of each strand is made up of sugar and a phosphate, and these two strands interact with each other through nucleotide bases. So a DNA is made of the 4 nucleotides. So now let's understand this in more in detail. A nucleotide is made of the 3 components, that is a sugar, that is deoxyribose, and then a phosphate group, then a nucleotide base. So these the nucleotides are joined to one another in a chain by a covalent bond. that forms between a sugar of one nucleotide and the phosphate of the next. So resulting it in to form an alternating sugar phosphate backbone. Nucleotide of two complementary strands attached to each other, and these are called as base pairs. So they attach to each other through a hydrogen bonds. For instance, we have the 4 nucleotide bases where adenine base paired with the thymine through the 2 hydrogen bonds, while cytosine is bind to the guanine through the 3 hydrogen bonds. So if a somatic mutation occur in a cell, single cell, in a somatic cell, that cell is progenitor of a population of identical mutant cells, all of which have descended from that cell get mutated. So a population of identical cells derived asexually from a one progenitor cell is called as a clone. So because the members of a clone tend to stay close to one another during development, so an observable outcome of a somatic mutation is often a patch of phenotypically mutant cells called a mutant sectors. So the earlier in the development the mutation event, the larger the mutant sector will be. So mutated sectors can be identified by the by eye only if the phenotype contrasts visually with the phenotype of surrounding this particular biotype cell lines. So these mutations can be acquired mutations that are most common specifically in case of the cancer. It could be a germline mutation that are less common specifically in the cancer. So now let's understand what is actually a mutation. So usually we know that DNA is incredibly stable molecule, allowing a genetic code to be maintained and ultimately passed on to the offspring. So despite this stability, there are some of the instances when sequences within the DNA can change. So that change is called a mutation. So the type of mutation can be characterized by the any change that it produces within the sequence. Like the point mutation is the result of a change in a single nucleotide. So in the point mutation there can be a substitution, insertion, and the deletion. In the substitution, when a one nucleotide is replaced with the other nucleotide. In case of the insertion, when there is addition of one nucleotide within the sequence. And deletion, it is the ultimately deletion of one single nucleotide from the sequences, that is deletion point mutation. So besides, so there are the mutations that are based on the impact they have on the genome. Like for the missense mutation, it is a change of a single nucleotide DNA base that result in a change of in amino acid sequences. So sometimes a single amino acid can change greatly or alter the protein function, or sometimes it doesn't. Then the nonsense, in case of the nonsense mutation, if a change in a nucleotide base that create a stop codon, so eventually it will terminate the translational process. Ultimately it will generate a immature protein or a abnormal protein. Then the frame shift mutation, in case of the frame shift mutation, it will overall change the sequence or the frame of a particular sequence, and eventually it will result in the change in the structure and the function of a protein. So a change even at a single nucleotide base can be good enough to change the type of the protein it expresses. So now let's understand how it happens. So as we know, each amino acid is represented by 3 nucleotides, and that is called as a codon. So mutation in a codon sequence can result in change to the amino acid it produces. So eventually once the amino acid is changed, so it will result the alteration in the structure and the function of the protein. So if you look at this table, here we will find how, like a triple T codon, if it getting mutated to the last nucleotide get mutated to A or the G, from the phenylalanine, now it will encodes for the leucine. So this show that if any change in a nucleotide, that can also result the can change the whole sequence of a protein. Eventually it will alter the structure as well as the function of a protein. So eventually, in case of the, let's understand how these mutations actually can impact the DNA, can enhance the cancer tumor formation. For instance, there are the three well-known genes that is called as tumor suppressor genes, and then the oncogenes or the DNA repair genes. So as all we know, the tumor suppressor genes are actually the normal function of these genes is actually they regulate the overall cell growth. So if there is any change or any mutation in these genes, so it will alter its normal function. So eventually the growth become uncontrolled and it will result in formation of the tumor. Then the oncogenes. So in case of the oncogene, the normal function of these genes are they are actually the normal genes that are involved in a cell growth or the cell cycle processes. So if any mutation that take place in these genes, so that will result the uncontrolled growth. And then the DNA repair genes, the normal function of these genes is actually if there is any change take place, these repair genes eventually repair those genes and repair that changes. So if any mutation take place that in the DNA repair gene, it will alter their function. So eventually it leads to the uncontrolled growth of the cells. Then on T-info platform, we have various kind of pipelines, including here we can also visualize how our reads are aligned to the genome. So like after once you start your pipeline, so then the on a butyl2G, here your genes will be aligned on a genome, and then you can visualize this. So these are different pipelines that you can explore for the on T-info platform. So the result of these visualization, here you will get like once you have able to align your reads on a genome, so you can visualize those genes, those alignment on a reference genome using various browsers. Like in our platform, we have already integrated JBrowse. So here, as we can look into this image, you will on the visualization you will get to directed to this platform. Here this is like a reference genome where your different reads are getting aligned. So then you are getting this peak. So this peak is actually indicating the coverage of the reads that are getting aligned to the reference genome. So higher the peak, it indicating that high is the coverage, but it means, so like here we are getting the in this particular region on a reference genome, they are the maximum number of the reads. That's why it is getting we are getting higher peak. So higher peak indicating there is high coverage. It also indicating there is high confidence of this particular region. So when there are then there is a region where you have less number of the reads, so you will get a very small peak or you won't get even a peak. So in that case, it indicating there is low coverage and the low confidence. So this is how you can visualize alignment of your reads on a reference genome using T-info platform. So then apart from the visualization, you can also perform various kind of mutational analysis on a T-info server. So like here we have a mutational pipeline. So here you will under the mutational section, so like here, so under the genomics section you will find a option section that is mutational variant or parallel analysis of mutation. So here you will find a pipeline for mutational analysis. So here once you align your reference your reads on a reference genome, then it will, there are the different number of the algorithms with which you can perform various kind of variant calling, or that is variant calling or the mutations. So then there is a differential variant calling. So like Strelka, it will produce, eventually it will compute the differential mutation between different groups of the sample. So with the somatic snipper, you will able to perform differential mutational analysis. And finally, you can end this pipeline. So you will find different pipelines at our T-Bioinfo platform to perform various kind of analysis on a genomic data. So in the mutational data, once you run this pipeline, so I just discussed here, it is difficult to demo every pipeline here in this one single session. So like in the after the running the differential pipeline, so here you will get this kind of output. In this output, it will produce a table like in which you will get the like chromosome position, then the position on a the chromosome position of that that particular position, then it will indicate the original reference genome, and then like here C's, then but kind of the mutation that is taking place in the tumor. So like here C is getting mutated to the C, and then like here G is getting mutated to the R. And then in the next column, that is where you will find the genotype of the normal sample that you have provided. So this is like the different groups that you are going to provide. So here, like in this case, in the normal samples, so G will remain in the G. So then the most important column that here you will get, that is somatic score. So somatic score is actually will indicate you the significance of your mutation. So if there is high mutation somatic score, it indicates that there is a high confidence that it is highly significant mutation that is taking place. And this is then the tumor consensus quality. And apart from, you will get actually detail of each column that representing. So these are very important columns that you are getting here, especially somatic score and the kind of mutation it is taking place. So you will get this kind of table after performing mutational analysis on T-Bioinfo platform. Then apart from that, you will get this kind of substitutions information as well. Like here, what A indicating and C indicating, T indicating. As here we already got this R, so what it does, it means, so it indicated that our initial G is actually now mutated to the AG, that is dinucleotide. So this is key substitution tables. You will get the even information what are these mutations. If apart from those 4 codons, if you get any kind of other, so you will get all those key substitution details of all those mutations. So then we have the 3 courses at our educational platform for the genomics data. So like introduction to the genomics and genomics 1. So introduction to genomics, where you will get the details, like the basic concepts and the principles. Then you will get details regarding various sequencing techniques, and you will get also the what kind of data analysis approaches that are available. So under the genomics 1, you will get details like the processing of genomic data, and under the next section, that is single nucleotide variations. And here you will also get the details how you can perform visualization of the mapping. So this is how you will here we are just giving you overview. So in these courses at educational platform, you will get details of all the topics that we have discussed in much detail. So then, apart from the genomics, proteomics or transcriptomics data, we have the another data, that is metagenomics. So the that data that we have discussed so far, that deals with the single organism. So what in real life, especially the organism live in a community and with in particular environment. So they interact with each other as well as they interact with the environment. So to study the community and look across the genomes of the organism living in, it is actually the branch that deals with it is the metagenomics. It can be defined as genomic analysis of microbial DNA from environmental communities. So metagenomics tools enable the population analysis of culturable and previously unknown microbes. This is important as only around 1 to 2% of bacteria can be cultured in a lab. So the ability to identify microbes without a prior knowledge of what a sample contains is opening a new door to various disciplines like microbial ecology, virology, microbiology, environmental sciences, and biomedical researches. So the sequencing based examination of these metagenomes has become a powerful tool for generating novel hypotheses. In the prokaryotes, there is the most important component that is used to sequencing and analyze the phylogenetic relationship between different organisms is 16S ribosomal RNA. So it is this 16S ribosomal RNA is a component of 30S small subunit of prokaryotic ribosome that binds to Shine-Delgado sequence. So the sequences coding for it are referred to as 16S ribosomal RNA gene, and they are used in the reconstructing the phylogeny due to the slow rates of evolution of the region of that particular gene. So the multiple sequences of the 16S ribosomal gene can exist within a single bacterium. So at T-Bioinfo platform, we have an option that is where you can perform metagenomic analysis using this 16S amplicon data. And you can generate the phylogenetic relationship between different organisms using various tools like phylosec and the ape2. So no, let's understand one term, that is OTU. I think most of maybe you know that. That is OTU is actually indicating operational taxonomic unit. So it is used to classify groups of closely related individuals. So classification can be based on different methods like hierarchical clustering, and then it could be a Bayesian clustering. So there are different algorithms based on which you can perform various kind of clustering analysis, and you can generate various kind of phylogenetic trees. So do we have time, Dr. Mohit, to perform, this demo of this particular session, Code OmicsLogic?
No, Dr. Preetan, it's over time, but I would like to ask Dr. Ramesh that.
Yes, sir.
Yeah, Dr. Ramesh. So is this like, I think we did covered a lot of technologies
It
like
was so
genomics.
nice.
Yeah, thank you.
Yeah, I think so everyone.
For,
yeah, I'm saying to a
yeah.
student only, sir. Actually, if you have any question or things, just try once. Actually, sir, the video already they will receive actually on mail.
Yes, they will receive.
Yeah, so
in the
again
mail.
at your actually you can say this is a homework. It means you have to do whatever madam told. Actually, we have to follow that process. Again, I did actually. I downloaded that notepad. Then we have to convert in that Excel format, and we have to make. You can at least up to that step. You can go ahead actually. Okay. So everyone, for this is homework.
And we are giving you. Okay, so just 1 second, Dr. Mohit. Just I asked two things so that before the next sessions, as today we know that you are unable to access some of the resources.
Yeah,
and
there are so many students actually. There are so many students got problem. So I hope so. They will receive suppose video. They have to follow it actually.
Yeah.
Okay, so then he will suppose they will come to know, and I am here only to if it's little bit problem I can help.
Okay, so before the next session, we just to ask you the few things so that you can complete it. So once you are with us, so that's why you able to understand things are better. So first thing you must do it, you complete your profile at educational platform that already I think Deepsa has already shown you. So then you also, if you you complete the introduction to the bioinformatics. So that is one free course available. And especially to understand the, to run a RNA-seq pipeline, you go through the transcriptomics 1 course. And if you are interested, then you, depending upon your interest, that is, you can go through the introduction to genomics or the metagenomics 1 course. And also run this demo pipeline for RNA-seq, and must go through the transcriptomics 1. That will give you a detailed and broader perspective and will give you a better understanding. And the next, you can skip that as we haven't explored the coding platform. So we will do that in the next, our next session. So now over to you, Dr. Mohit. And
Yeah,
if you
thank
have
you,
any
Doctor.
questions.
So
Yeah, I
profile,
will
madam, I
address.
think so everyone completed. I received their profile link.
So
Most of all, they have sent me. So I made in that Excel,
so
sir, whatever I sent now, that Excel. In same, actually, next column, I have given that link. So I'll send you today. Some faculty also, they left actually. So I'll tell to that faculty. Because faculty, everyone having their so busy schedule now. So that is
right,
the,
right. So Dr. Ramesh, let me explain few things which Harpreet showed. So Harpreet, I'm
okay,
sharing
sir.
my screen. So this is the page. So you will find the video recording of this session and everything here. So if without, even without emails, even if you don't get an email, you will be able to get it in this page, and everyone have access to this page. So on the educational platform, you have profiles. So I will just show you. This is the page, right? And Deeksha has shared the link, and she'll share it again. So Deeksha, please share this link. So this is the forum, and here if you see, Deeksha has already posted about the Python thing, and R and Python and installation, and here you will see, you will be getting updates from all of the activity that is happening. So if Dr. Ramesh is doing a course, if you are finishing a course, it will show here. And by completing a profile, you have to complete it like entirely. So there is no profile picture other than Shreyash T. So let me open Shreyash's profile, but this is also not complete. So what does that mean? So this is very important. Always keep your first name and second name separate. So Sikandar space Mula, if I'm right. Veera under space Raghavan. So this is one important thing. Another important thing is to fill out this information that you are from REVA University, and other details. So you can do one thing. You can go to your profile here. If you click on your profile, you can go to this tab, and then you can edit different things accordingly. So you can put up profile picture, cover image, you can put, and you can write little things about yourself, where we will know that you are from REVA University School of Biochemistry. So all those details, please insert this into your profiles. Right now the profiles have been created, but they are not complete, because you see here so many people have joined, but the images are no photographs, and there is no details. So please complete that. So that's what Dr. Harpreet meant, that complete the profile. And in this page only, you will see different things, right? Forum, courses, projects. So I just refreshed the page. So once you refresh the page, you will see more updates. Like people, if you're changing the profile picture right now, it will show. If you're doing something, it will show. So that's why how we see that everyone is doing something or progressing. So forum. So forum is this. Here we will have a session recording. This is a session recording topic. In this topic, you will find all the video recordings. So we will be posting it here, and I'm again sharing it in the chat so that you can see this. So here we will be posting the video, so you will get it directly, and we will also email you. So you will also get it in the email. So there is no problem, I guess, to access this. And this another tab is courses. So the assignment is it's very important, and please note, all of you should finish Introduction to Bioinformatics, and then we discuss Transcriptomics 1 today and Introduction to Genomics today. So if you want to finish Introduction to Genomics, please go ahead. But Transcriptomics 1, that will be discussing in more depth. So please take your time. and start exploring transcriptomics 1. Transcriptomics 1, genomics, and bioinformatics. Similarly, you have all these courses. So it's up to you. Introduction to metagenomics is also, that's what we covered. But the first step is to complete introduction to bioinformatics, and then transcriptomics 1 or metagenomics or genomics. So there will be a specialization that you would, if you're interested in DNA variation, you go for genomics. If you are interested in, gene expression, go for transcriptomics. If you're interested in, viruses and small bacteria and all those, then it's metagenomics. Environmental sampling, then it's metagenomics. So you can choose a specialization here, but we will be talking in the context of, data science and Python and R, and in the next session we will have demonstration of the code platform. So it would be nice for you to understand the basics. So that's why we covered the basics today. And look at the video again, and look at the courses. Everything is in the courses. So the courses, if I open this, let's look at how a course is and how we can take this course. You can easily take the course. It is made available for you. So please do this, because it will be available for 1 month. And I think, as Dr. Ramesh said, make the most out of it. And this is a typical course. You have an overview, what it is about. You can see a video also on the top of things. And then there's a curriculum where you will be, when you are completing any of these sections, it will be coming up as like this, a tick mark. So once it is tick mark, it means that you have completed. So once you have completed the entire course and have taken the quiz, you will obtain a certificate for taking this course. So every course that you take, you will get additional certificate, and also you will get a certificate after finishing this program. So there is a lot of certificates to earn from, and there is a lot of content to look at, and it's not very difficult. It's a course that might take you maybe 1 hour. Course which tells you about the bioinformatics in healthcare, translational bioinformatics, agriculture, environmental science, defense, public health. So personalized healthcare. So if you're interested in any of these domains, it will be really helpful for you to understand the application of bioinformatics. So introduction to bioinformatics, please finish this. And in the next class, we will review how everyone is doing, and we will point out if you're not finishing. So it's a homework, and we will.
Sir, you will track now whomever student, whomever they are not doing performing, and how their actually activity. You can find out, sir. Actually, I will
everything,
see
everything. As I was telling you, this progress. Yes, I will share the detail. We will start next session tomorrow 9 a.m. and before during the session we will talk about the progress. So we will look into every profile, and I have shared Excel file with Dr. Ramesh and HOD Dr. Jayashree. So you can, if you don't have the password for your TIBA Info platform, please reach out to Dr. Ramesh, and he will tell you the password for your account. So it's with Dr. Ramesh also. You can ask us also, like Deepsha at the marketing@pine.bio. So if you have any difficulty, any issue, you can just write it here in the forum, that create a new topic. So this is like a topic. If you want to say that, where is the video of today's session? So you can write this here, and you can just simply submit. Similarly, this forum has different topics. You can create your topic. You are having problems, you write it here, and we will address it. So you will have the support, and you will have access to all the material. Now you just have to start learning, and that's all. And I'll pass it on to Deepsha now to conclude the session.
Thank you so much, Dr. Mohit, because everybody is having some kind of questions regarding all the different resources. I just wanted to show you the email that has been sent to you. So as Dr. Mohit was describing to you, you have 3 different accounts, and they are all linked to your REVA ID. So the first one is the Edu account. The Edu account is the one which many of you may have created. And if you have not created that account, we have sent you an email ID, a username and password for it. So the email username will always be your email ID. And we have generated a password and sent to you. But many of you, like as far as I know, like Akshata, like I think even Shreyashi and a lot of other people's already had their own Edu accounts. So you can log in with the same email ID. Okay. The next is the tbioinfo account. So this is the link to the server tbioinfo where you will have the details. So you cannot create an account on server tbioinfo. We have, that's why, created a username and password. Whenever you log into server tbioinfo, it will show you the username as in capital. All the letters will appear capital. That is perfectly all right. And that makes the server more secure. So that's why this username, and it is going to be the same REVA email ID, because your email ID is your identification. It is linked to all of your accounts. Again, this is generated for you. So this is the server password from where you will be able to do the demo pipeline. And the third is the OmicsLogic platform. We cannot generate password for you for this OmicsLogic. You have to register and choose the starter pack and then apply the code as I had shown at the beginning of today's video. So please review that again. Use your same REVA email ID. Go to codeomicslogic.com, click on pricing and add this code, and you will be prompted to add your card details, but your card won't be charged. So if you have any questions regarding these 3 accounts, please let us know. Email me anytime, marketing@pine.bio, for all these details so that you do not face any trouble. And by the next class, we are all ready with all the different access. So please let me know throughout today if you have any kind of questions. And you won't be able to use these username and password on this platform. Like, you can definitely create them. So you have to create separate IDs. So these are all 3 separate accounts. And you will just have to create IDs for this account now, if you already have the Edu account, which most of you now do. So Edu tbioinfo is done. OmicsLogic is left, and we will be covering OmicsLogic in the next class. So thank you so much for joining today. Do you have any more questions?
So there is one question Shreyashi Banerjee is asking. So she's saying, sir, under courses it's empty now. After taking up, will it appear in that block? So I'm not sure what.
Mohit, we will. I will ask Joshua to look at it, because everybody is being enrolled, so it is okay.
Okay then. Thank you everyone for joining, and I hope that Dr. Ramesh and everyone that you had a we had
one thing, sir. One question, sir, for my side. So very basic question, sir.
Sure.
So what we are saying actually, read. Okay, that is very small size read and medium size read, and somewhere read it was very high peak. Actually, we are saying this is a more confident read is there. So what is the how we can understand a very easy way that read? So this is a I can say a particular sequence having very good sequence, or you can say how you can understand very easy way in read.
Read means re-representing the sequence. So the high coverage that I was saying, so it indicate the high confidence when we align on a sequence. So we are letting you know that the high coverage means the if you will get the more reads for that particular reason, right? The number of the reads are maximum number of the reads that getting aligned to that particular reason. So it will tell you it with the more confidence we are saying that these reads belong to this particular genomic region. I hope it clear to you now.
No, somewhere, madam, it is a small read means when we will do the sequencing means all this data. So this is a small fragment. They will give then big some medium size read. So or the means on the basis of size only they are making, or how we can say it?
when we are saying small or the large reads, that
Yeah,
is
yeah.
the length of that we are saying kind of length of the sequence. But when we are saying high coverage, that is in terms of coverage, that is it giving the confidence that this read is actually belong to this particular region. High coverage and low coverage is the confidence that we are saying that these reads belong to this genomic region at a reference genome, right? And length of the that when we are saying small and the long reads, that is we are talking about in terms of the size of the reads. Small
Okay,
reads
okay.
usually belongs to like 30 to 100 base pair, right?
Yeah, yeah,
So
yeah, yeah.
like long reads means it
So,
belongs to that it contains large length of the sequences. It's clear to you now?
yeah, so different
Yeah.
machines, different reads. So, right, Harpreet? So that depends on the experiment that we are trying to conduct. So if we are looking at the, entire genome,
There
so
are no,
we
sir,
will,
some means like that some what you can say parameters should be there now. How much read we have to keep actually? We have to set fix actually. Read should be supposed 2 KB or 1.5 KB read. It
no,
will
no,
be good
that's
first.
what that's what I'm saying. So there are long reads, there are short reads. So based on the experiment, like sequencers, it's like some sequences are giving short reads, and so you design your, experiment based on that. And once you run the experiment, then you get reads. So you cannot decide, like, the kind of coverage that you will get. That depends on the sample, how clean it is, and if there are technical problems, and if there is like this is the entire process of NGS experiment. So from the experiment, you're getting reads, and now after the experiment, you have the reads. Now you have to understand how to analyze it. So that's what we are doing. So we are not doing the experiment. We are just taking the data that has come from the experiment and now analyzing that data.
Okay, sir. Okay. 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 Thank you
And
very much.
Dr. Mohit,
Even the detail,
Okay.
I just want to add one point. So the details of all those things, I mean, you can find under the course, Transcriptomics 1 course, as well
Yes.
in the some of things that you will find even the genomics, under the
I'll go
genomics
through
1
your
course.
paper, ma'am. I will see. I'll try to
Yeah,
learn.
yeah. You just go through those courses.
Yes. Yes.
So you will find the more details regarding, I mean, that will, I believe that those will address your, I mean, deep concerns like
All the
these
issues.
questions.
Yes. Yes. Absolutely. Every basic
Okay.
concept is well described, and any student and anyone can learn. You just have to go through this simple courses and let us know if you have any problem.
yes, thank you, Dr. Mohit and Dr. Harpreet, and also Dr. Ramesh. I would want to add again, because of the questions that we are getting right now. So the OmicsLogic is a separate platform from the Edu platform. So you will need to create an account again. So just register with the email ID, the same email ID.
Deepsha, can we just have the code platform tomorrow? So let's just focus on server and Edu, and we will do the code platform tomorrow. So don't go into code platform right now because we did not cover it in today's session. So please make your Edu platform ready and try accessing the server platform, and we will discuss the code platform tomorrow. We'll start with that, and we will see that if you have access and you can log in and stuff.
Yes, thank you, Dr. Mohit, for this, and also for the session recording. So right now, the session is being recorded. It will be processed, and I will share it on the forum under session recordings. So the link to the forum is given right now in the chat. Similarly, I will be sharing this via email with everyone. So please look out for that email again. And soon as it is processed, we will be sharing it with you. And please let us know if you have any other questions in the forum itself. So thank you for joining us today. We will meet you all tomorrow again at 9 a.m.